Okay, we are actually in chapter 8, which if you might think that's a little bit weird because we were just in chapter 5, and it's true, we skipped 6 and 7 for now. We're going to come back to them after this, okay? Chapter 8, go back to 6 and 7. Chapter 8 is about DNA. Now here's the thing. Up until this point, actually chapter 6 and chapter 7 are about genetics. So what is genetics? So it is about heredity. So scientists actually, before DNA was discovered, or before the structure of DNA was discovered, scientists already knew a lot about how heredity. So in other words, how traits are passed along. Heredity, which is how traits are passed along, like dark hair, eye color, right? All that kind of stuff. How traits are passed from parents to offspring. And some of you might have done these things called Punnett squares in middle school, right? With the the different alleles, like a capital A and a small a and a capital B and a small b, and you cross them over. So, see, those Punnett squares were invented a long time before DNA was understood, okay? So people knew a lot about how things were passed along from parents to offspring, but they didn't know what chemical carried the information. So listen, this is important. They knew that there must be some chemical in the cell, some molecule that is being passed from parent to offspring that has the information in it about eye color. I mean, obviously, parents don't pass along an actual little tiny copy of themselves to their offspring, a little person, right? But what it is there must be some kind of a code, some type of information that is being passed along. They didn't know what it was. Now, you've already learned about some of the different molecules. So, for example, I could ask this question. Was it a protein? Because there's all kinds of proteins in the cell, right? Was it a carbohydrate? Was it a nucleic acid? And that's where some people thought the clue was, or that's what they were focusing on. That's what Francis Crick and James Watson kind of assumed it was the DNA. Why would they assume that? Because it's in the nucleus, all right? And people knew about chromosomes and stuff, so they know it's part of the chromosome. Of course, the chromosome also includes protein. So see, you're not entirely sure. Even if you know that this is in the nucleus and it's going to get divided up when the cell divides and everything, you're still not entirely sure. So they wanted to know, what is that thing that's being passed along? There were actually 3 experiments. 3 experiments before Watson and Crick that indicated or suggested that genes were made of DNA. What is a gene? It's the genetic information. So let me point to genes here. This is the genetic information. This hereditary information. That information is made of DNA. There were 3 experiments, so let's talk about each one in turn. Now, this is what this next assignment 21, is going to be about, is this first section of chapter 8, section 8-1. It's about these 3 experiments. 8-1 does not actually cover what you just watched. 8-1 is before that. The first one is an experiment that is just called Griffith's experiment, and it's actually pretty cool. The first two experiments involved mice. Griffith's experiment, 1928. When was that movie? Did you guys catch that?
It was in the 50s.
50s, right. 1962 is when the prize was awarded, right? And they were mid-50s or so when they discovered that that structure. So here's what he did. He took some mice. Now, he started off, and this diagram is in your book on page 172. All right. So I'm going to kind of reproduce the diagram here, but you can look at it in your book as well.
Is this being recorded?
Yeah. So he started off. There's two kinds. He used two kinds of bacteria. There's a lot of kinds of bacteria, obviously. He had two kinds, two particular species of bacteria. One was smooth. In other words, if you looked at it under a microscope, it had a smooth, or actually it made smooth colonies on the petri dish. We're going to do an experiment in several weeks where we grow bacteria, and they make these little spots on agar and this kind of jelly stuff that you grow them on, and they make little spots as they grow colonies. And the smooth ones made smooth, round colonies. So one was called smooth, and it was deadly. It was a disease bacteria. The other one was what they called rough, and it actually made rough-looking colonies, like the colonies that it made on the petri dishes didn't have smooth outlines. They kind of had rough outlines. So it was called rough, and it was safe. So here's what you do. You take two mice,
What is that?
okay,
It's adorable.
and you take, all right. Now, by the way, when they use mice, one of the things, after you did the onion thing, remember how we tried to keep the onions the same size? One of the things they do with lab rats or mice is they try to breed them so that they're almost genetically identical. They do that by inbreeding, actually, to almost make clones of the mice so that they have all identical attributes. Because you don't want one mice to be really strong, right, and one mouse to be weak. So they want them all to be the same. Anyway, you inject them, okay? So we're going to take a syringe, and we're going to inject the mice.
The second one doesn't have to.
We're going to inject the mice with, this one's going to get injected with the smooth bacteria, and this one's going to get injected with the rough bacteria. So one's getting injected with the disease, the other one is not. This one is dead, okay? And this one then, whoops, this one is the same. This one's fine, okay? Now here's where that was not the interesting part of the experiment. That was obvious to everybody. They already knew that was going to happen, okay? But this is just kind of a control. This is a control. They're testing out the two bacteria beforehand. But then what they did was they took some of the smooth. They killed the smooth bacteria with heat. So they put it in a test tube and they heated it up over a Bunsen burner so that it killed it. Then they injected the killed bacteria into a mouse. I'm actually going to run out of time here, aren't I?
Told you.
They injected the killed bacteria into the mouse, and the mouse stayed alive. So in other words, if the bacteria is dead, it doesn't kill the mouse.
Well, yeah.
Okay.
Isn't that kind of obvious?
And I guess I'll save the punchline for next time.